Imagine a world where extinct creatures roam the Earth again. This isn't science fiction, it's a burgeoning reality. Thanks to groundbreaking advancements in gene editing, the possibility of bringing back creatures like the dire wolf is closer than ever before. Gene editing, particularly through technologies like CRISPR, acts like a tiny pair of scissors, allowing scientists to make precise changes to an organism's DNA, its genetic blueprint. By leveraging this technology, scientists believe they can rewrite the DNA of modern canines, transforming them to resemble their long-extinct ancestors. The dire wolf, a formidable predator and a larger, more powerful cousin of the gray wolf, roamed the Earth thousands of years ago before vanishing into the annals of history. However, their legacy lives on through the remnants of their bones and teeth, which still contain traces of their ancient DNA. This ancient DNA is a treasure trove of information holding the key to potentially recreating the dire wolf. By meticulously comparing this ancient DNA to that of modern wolves, scientists can pinpoint the genetic differences that made the dire wolf unique, paving the way for its resurrection. The prospect of resurrecting extinct species is both thrilling and daunting, presenting a blend of excitement and ethical dilemmas. Imagine the awe-inspiring moment of hearing the haunting howl of a dire wolf echoing through the forests once more, a sound that hasn't been heard for millennia. Achieving such a feat would be a monumental testament to human ingenuity and scientific progress, showcasing our ability to unlock the secrets of the past. Yet this scientific marvel also raises profound ethical questions about our role in the natural world. Should we bring back species that nature has selected for extinction? What are the ecological implications? These are questions that scientists, ethicists, and society at large must grapple with as we stand on the brink of a new dawn for the dire wolf. At the heart of this scientific revolution lies CRISPR-Cas9, a revolutionary gene editing tool. This tool has opened up new possibilities in the field of genetics, allowing scientists to make precise modifications to DNA. Imagine DNA as a long chain of letters like a genetic code. Each letter represents a specific instruction that determines the characteristics of an organism. CRISPR-Cas9 acts like a pair of molecular scissors that can cut this chain at specific points. These cuts allow scientists to remove, add, or alter sections of the DNA sequence. This ability to target and cut DNA with such precision is what makes CRISPR-Cas9 so powerful. It's like having a highly accurate tool that can make changes at the molecular level. Think of it like editing a text document. Just as you can use a word processor to find and replace words, CRISPR-Cas9 allows scientists to find and modify specific genes. CRISPR-Cas9 allows scientists to pinpoint specific genes within an organism's DNA, just like you might use the find function in a word processor. This precision is crucial for making targeted changes without affecting other parts of the genome. Once they've found their target, they can then use the cut function to remove or replace specific genes. This process can be used to correct genetic defects, enhance traits, or even create entirely new characteristics. This technology has the potential to revolutionize medicine, agriculture, and even conservation. In medicine, it could lead to cures for genetic diseases. In agriculture, it could create crops that are more resistant to pests and environmental stresses. In conservation, it could help save endangered species by correcting genetic issues. It's like having a genetic toolkit that allows us to make precise changes to the building blocks of life. With CRISPR-Cas9, the possibilities are endless, and the future of genetic research looks incredibly promising. The journey to resurrecting the dire wolf begins with finding its genetic blueprint. This blueprint is hidden within the ancient DNA of the dire wolf, a species that roamed the Earth thousands of years ago. Luckily, traces of this blueprint still exist in the form of ancient DNA. These genetic remnants are like time capsules preserving the biological information of the dire wolf. This DNA can be extracted from well-preserved remains such as bones and teeth found in permafrost or caves. These natural environments act as freezers, slowing down the degradation process and keeping the DNA intact for millennia. These ancient DNA fragments are like puzzle pieces scattered across time. Each fragment holds a part of the genetic code, and scientists must carefully piece them together. Scientists use sophisticated sequencing techniques to piece together these fragments, gradually reconstructing the dire wolf's genome, its complete set of DNA. This process is akin to assembling a complex jigsaw puzzle without a reference picture. This reconstructed genome serves as a roadmap, guiding scientists in their quest to edit the DNA of modern canines. By comparing the dire wolf's genome with that of living species, researchers can identify the unique genetic traits that defined the dire wolf. However, working with ancient DNA is like trying to read a faded and fragmented scroll. The passage of time has not been kind to these genetic materials. The DNA is often degraded and contaminated with genetic material from other organisms. This contamination can come from bacteria, fungi, or even the soil in which the remains were buried. This makes it a challenging and meticulous process requiring advanced techniques and expertise. Scientists must carefully separate the dire wolf DNA from these contaminants to ensure the accuracy of their reconstruction. Once scientists have a good understanding of the dire wolf's genome, they can begin the process of editing a modern canine's DNA to resemble it. This involves using CRISPR-Cas9 to make precise changes to the canine's genes, essentially rewriting its genetic code. Imagine taking the DNA of a gray wolf, the dire wolf's closest living relative, and modifying it step by step. Scientists would target specific genes responsible for the dire wolf's unique traits, such as its size, skull shape, and bite force. By carefully introducing these genetic changes, scientists could potentially breed canines with more and more dire wolf characteristics over multiple generations. It's like sculpting a living organism, using genes as their chisel. Section 5. The ethics of resurrection. The possibility of bringing back extinct species raises profound ethical questions. Should we interfere with the natural order of life and death? What are the potential consequences of reintroducing extinct animals into the ecosystem? Some argue that de-extinction could help restore balance to ecosystems that have been disrupted by human activity. Others worry about unintended consequences, such as the spread of diseases or the displacement of existing species. The debate over de-extinction is complex and multifaceted. It requires careful consideration of the scientific, ethical, and societal implications. We must proceed with caution, engaging in open and informed discussions about the potential risks and benefits. Section 6. Navigating the maze. Bioinformatics and de-extinction. The sheer volume of data involved in de-extinction projects is staggering. Sequencing and analyzing ancient DNA, comparing it to modern genomes, and designing precise gene edits require immense computational power. This is where bioinformatics comes in. Bioinformatics is like the compass and map of the genetic world. It uses computer science, statistics, and mathematics to make sense of complex biological data. Bioinformatics tools help scientists analyze and interpret vast amounts of genomic data, identifying the key genes involved in specific traits. Without bioinformatics, de-extinction projects would be like trying to navigate a labyrinth in the dark. Bioinformatics provides the light and guidance needed to make sense of the complex world of genetics. Section 7. IT, the unsung hero of the genetic revolution. Behind the scenes of every groundbreaking scientific discovery lies a powerful engine information technology, IT. From storing and processing massive genomic datasets to running complex simulations, IT infrastructure is essential for de-extinction research. High-performance computing clusters, data storage systems, and sophisticated software are the unsung heroes of the genetic revolution. They provide the computational muscle needed to analyze ancient DNA, design gene edits, and model the potential consequences of de-extinction. Just as a powerful telescope allows us to see deeper into space, advanced IT infrastructure enables us to explore the vast and intricate world of genetics. It's a vital tool that empowers scientists to push the boundaries of what's possible. Section 8. Beyond de-extinction, a future shaped by gene editing. While the prospect of bringing back extinct species captures our imagination, gene editing has the potential to do so much more. It holds the key to curing genetic diseases, developing new crops resistant to climate change, and even enhancing human capabilities. Imagine a world without genetic diseases, where inherited conditions like cystic fibrosis and Huntington's disease are a thing of the past. Gene editing could make this a reality by correcting faulty genes, offering hope to millions. In agriculture, gene editing could lead to crops that are more nutritious, resistant to pests and diseases, and better able to withstand the challenges of a changing climate. This could help us ensure food security for a growing global population. Section 9. A call for responsibility. The power to edit the code of life comes with great responsibility. As we venture further into the uncharted territory of gene editing, we must proceed with caution and careful ethical consideration. Open and transparent discussions about the potential benefits and risks of gene editing are crucial. We must establish clear ethical guidelines and regulations to prevent the misuse of this powerful technology. The future is in our hands. By embracing both the potential and the responsibility of gene editing, we can harness its power to create a healthier, more sustainable, and more awe-inspiring world for generations to come.